DNA stands for deoxyribonucleic acid. DNA is a long, thin molecule made up of nucleotides. There are 4 different types of nucleotides. These are adenine, thymine, cytosine, and guanine. And each is represented by their first letter, A, T, C, G. DNA is like a recipe that holds the instructions for telling our bodies how to develop and function. Your cells endure DNA damage thousands of times each day. Given that your body is made up of around 100 trillion cells, that equates to a mind-boggling number of DNA errors, potentially up to a quintillion each day. Since DNA carries the essential instructions for building proteins that ensure your cells function properly, any damage can lead to serious issues like cancer. DNA errors come in different forms. Sometimes the building blocks of DNA, known as nucleotides, suffer damage. Other times, nucleotides get mismatched, creating mutations. Occasionally, breaks in one or both strands of DNA can disrupt replication or cause segments of DNA to become scrambled. Fortunately, your cells have sophisticated systems to handle these issues. Specialized enzymes are responsible for repairing various types of damage. For instance, during DNA replication, the enzyme DNA polymerase is supposed to match bases, adenine with thymine, and guanine with cytosine. However, it occasionally makes mistakes, inserting the wrong base. Luckily, this enzyme detects most errors quickly, removing and replacing the incorrect bases. A second set of proteins then reviews the DNA to correct any remaining mismatches. This process, called mismatch repair, reduces errors to about 1 in a billion. DNA can also suffer damage from chemicals, whether from environmental sources like tobacco smoke or naturally occurring substances like hydrogen peroxide. If a base is damaged, the cell uses a process known as base excision repair. An enzyme removes the damaged base, and other enzymes trim and replace the surrounding nucleotides. Exposure to ultraviolet light can cause adjacent nucleotides to stick together, distorting the DNA structure. This kind of damage is addressed by nucleotide excision repair. In this process, a team of proteins removes a segment of about 24 nucleotides and replaces it with new ones. High-frequency radiation, such as gamma rays and X-rays, can cause more severe damage by breaking one or both strands of the DNA backbone. These double-strand breaks are particularly dangerous and can lead to cell death. To repair these breaks, cells use 2 main methods: homologous recombination and non-homologous end joining. Homologous recombination utilizes a similar, undamaged DNA segment as a template to accurately repair the break. Non-homologous end joining, in contrast, trims and fuses the broken ends without a template, which can lead to errors but is useful when no similar DNA is available. While DNA changes aren't always harmful, some mutations can contribute to evolution. Maintaining DNA integrity is crucial. Defects in DNA repair mechanisms are linked to premature aging and various cancers. So, your cells are continuously engaged in an intricate repair process, working tirelessly to keep your DNA and your health in check.